Greetings, ladies and gentlemen. First, I want to thank the organizers for giving me the opportunity to present to you today, but also to apologize for the fact that a pressing, unexpected commitment has precluded my being with you here today in person. And so I appreciate your indulgence in allowing me to proceed with this talk by this video. As shown here on this first slide, I would like to take a perspective with this talk of a 40,000-foot look at the scientific journey, or as it were, the history of an HIV vaccine endeavor. I'm directing my talk to both the B-cell people, the vaccine people, as well as those that overlap both of those areas of endeavor. But I particularly want to address the younger fellows in the audience who may not have had the opportunity to experience the extraordinary history of this rather arduous road that we're on towards an HIV vaccine. So let's start off first, and I cannot let this go by without paying tribute to my long-term and dear friend, Max Cooper, whose 50 years of groundbreaking science we are celebrating here today in Banff. The picture on the left, I know that several of you have already seen, as Max in 1965 at the time of his seminal discovery of B cells. And on the right, Max today still expressing that keen intelligence, that excitement, and that enthusiasm for science that he had 50 years ago and that he maintains to today. So congratulations, Max. It's been an extraordinary pleasure being a colleague and friend of yours for so many years. So let's get on to the talk. I'm going to take a look at our approach to vaccine from two fundamental approaches shown on this slide: empiric or inductive, as I call it, and theoretic or deductive. Let me explain what I mean. When one takes an empirical or inductive approach, you're testing a candidate until a protection is achieved. Then you go back and identify the correlates and optimize the regimen to get the best possible result. On the other end of the spectrum is what we would call a theoretical or deductive approach, in which you determine beforehand the optimum immune correlate of protection, and then you design your immunogen to induce the immune correlate in question. Both of these are aimed towards an effective vaccine. Now let's take a look both historically and in reality for today of what these two approaches mean. Starting off with the empiric or inductive approach, this is what we have been doing classically throughout the years of what I refer to as classic vaccinology. The response to natural infection provides the initial proof of concept and the basis for the feasibility of our success. So let's go through that really quickly. So natural infection induces protective immunity. The correlates of protective immunity may or may not be known. In fact, among the many successful vaccines that we do not have precise correlates of immunity, but we try to get them anyway. You fashion a vaccine to mimic natural infection, usually using an inactivated or a live attenuated pathogen. The vaccine induces protective immunity, and then you try and determine, do you or do you not have correlates of immunity, which may not be even relevant because the vaccine, in fact, did protect. The examples of these are many. I show them on this slide: polio, smallpox, mumps, measles, all of which nature has already shown us that despite morbidity and mortality in some of these diseases to variable degrees, the body inevitably clears the virus in those who do not die and goes on to give you long-lasting protective immunity against the virus in question. Now, what are we dealing with today? Here is the first glimpse of what we were to see over the following 33 plus years. The introduction of HIV/AIDS, and in this case, before we even knew it was HIV, the initial reports of gay men with pneumocystis, Kaposi sarcoma, and other opportunistic infections. Within a couple of years, the virus was discovered first by the French, Barré-Sinoussi and Montagnier and colleagues at the Pasteur, and then a year later, the definitive proof by Gallo that indeed HIV was the causative agent of what we were calling AIDS. Very soon after that definitive proof, as shown on this now rather well-known statement by Margaret Heckler, which I'm going to get back to in a moment, where she said, we hope to have a vaccine against AIDS ready for testing in about 2 years. People ascribed to her saying that she said we would have the vaccine available, but she never said that. She said ready for testing, despite the fact that the other statement has been attributed to her. However, we had some complications. This is a paper from back in 1986 by Walt Dowdle, when he was at the CDC, who said that in our search for an AIDS vaccine, neither an attenuated nor a whole killed virus would be acceptable due to the undesirable presence of genetic material. In essence, live attenuated or killed was unacceptable for vaccine design, despite the objections to that concept by many. And so the spectrum of modern vaccine strategies were put before us, and the first that was done was the recombinant subunit vaccine. However, what we did not realize about HIV infection in the mid-1980s, at the time where we were going to and did engage upon the classical empiric approach towards vaccine development, what we didn't know that HIV was very different from any other virus for which we have dealt. The disease was almost invariably progressive, despite a measurable ongoing immune response. The natural immune response to HIV is inadequate to fully suppress or clear the virus. We did not know that because we weren't able to even measure virus early on. Neutralizing antibodies against primary isolates rarely occurred and certainly were not induced by vaccines. And the thing that really was a punch at us was that superinfection occurred despite an ongoing immune response against the autologous virus. With that as a background, not too different from what Margaret Heckler had predicted in 1984, that 3 years later, the first clinical study of an AIDS vaccine to be approved by the FDA was conducted at the NIH with the microgenesis GP160 subunit vaccine candidate. Now, over the next subsequent years, one can summarize the findings of a variety of phase 1 vaccine trials using these recombinant envelope immunogens is that, one, a majority of the recipients did indeed generate antibodies. Only a small minority of these antibodies were neutralizing. But most depressing of all, the antibodies that neutralized lab-adapted strains, but not primary isolates. This, in fact, had some icons of our time, like Dr. Maurice Hilleman, who was completely perplexed, and in this quote from a New York Times article in 1988, he said, I don't know anything like this, and he's had 45 years of experience in developing vaccines, that this is the first time in vaccine development that neutralizing antibodies don't seem to amount to much. We don't do them, we don't get them, and when we do, they don't neutralize the right strains. Another thing that was a little bit depressing to me, this is a picture of one of my mentors and a dear friend, Albert Sabin, who back in DC at a reception for both he and I at the time, whispered in my ear, and the truth, he says, Tony, I don't think we're ever going to have an HIV vaccine. Now, the reason for that was complicated, but we're going to get into that in a moment, and I will, when we get towards the end of the talk, I'll reflect back on both what Albert Sabin and Maurice Hilleman were saying. So over the years then, again, which some of the younger people in the audience will not appreciate, there were major disagreements and tensions within the scientific community concerning how do you approach an HIV vaccine. There was disagreement about the value of animal studies, about the advisability or not of doing many simultaneous phase 1 studies. disagreement regarding the criteria for launching into large phase 2 or 3 clinical trials, and always a concern about the cost of large studies versus the money being given to grants for basic research. There was an uneasiness in the community about getting going, and this is a typical quote from Don Francis, who was a major advocate of pushing ahead. He said, with 40,000 new infections in the United States each year, this is not something you sit around and contemplate your navel about. Now, there was a lot of disagreement about moving ahead, such a disagreement about whether or not the NIH, NIAID would fund large-scale studies of recombinant GP120 proteins. So there was a fierce debate, as shown by this article in the Philadelphia Inquirer. Should officials give the go-ahead or not to large-scale human trials or wait for something more promising? So I put a panel together, reported here by Larry Altman in the New York Times, to get members to look at the cost, the uncertainty that might be afforded, and to decide whether or not that we should, in fact, go ahead with the clinical trials. And what they said is that the uncertainty about whether enough volunteers could be recruited, the uncertainty about the protection, and the fact that members of the panel cited the large cost, they rejected the wider testing. Now, there were many, many cameras there. There was press all over the place, and this was going to be a big thing because this was quite a contentious argument in the public. However, something happened which keeps telling me that fate is much more powerful than your planning, is that just at the same time as we came out with this momentous decision, something happened in the real world, and that is O.J. Simpson, killed or not, his girlfriend and a friend, and was being followed by all the TV cameras on the Santa Monica freeway, and no one paid any attention at all to the fact that we made the decision not to go ahead with the vaccine trial. However, that did not at all deter individuals who decided they wanted to go ahead with vaccine trials. And in fact, a number of trials, 2 in particular, large efficacy trials, were launched, although many members of the scientific community felt that this was actually futile. In fact, at least with regard to the results, they were right, because both trials failed. A trial that was conducted in the USA, Canada, Puerto Rico, and the Netherlands, and one in Thailand in an antibody-based vaccine trial. So then the field decided that they needed another approach. We had been looking at B-cell-based HIV vaccine approach that was looking at preventing the acquisition of infection, even though we still were not successful in inducing neutralizing antibodies. And the field switched over, actually, to what was called the T-cell-based HIV vaccine approach that was looking not to prevent acquisition of infection, but to control the viral set point following infection. And we went for a number of years with extraordinarily elegant and productive studies in the animal model, in which T-cell-based vaccines, in fact, were able to control, as it were, the viral set point and even the peak of virus. And in fact, showing you some data for the slide for the paper on the right-hand side of the slide, you can see that peak viremia and viral set point in the vaccine-induced individual versus the control set point was significantly different. However, it was very clear that these effects did not last because, as you would expect, there were escape mutants. This was a T-cell-based approach, and sooner or later, the animals went ahead with disease. Now, we went ahead with some human studies. And in fact, we did a T-cell-based approach in now well-known studies, the STEP and Fambili studies, which were actually discontinued because the DSMB determined that this particular approach did not prevent HIV infection, nor did it affect the course of disease. However, there was some additional bad news associated with this, is that we found out that the T-cell approach, at least in this case, actually enhanced the risk of HIV acquisition in the vaccine group compared to the control group. And this allowed us to delve into the potential mechanisms that immune activation by a vaccine needs to be balanced by its ability to protect. So if a vaccine does not give you protection, it will inevitably give you immune activation, and you might have the paradoxical effect of actually having an increase in acquisition, which is what we saw. So nonetheless, the need, the desire on the part of some, with good rationale on the part of those who make the decision, but some disagreement on others who did not, went ahead with a proposed trial, and the proposed trial was to take two candidates that individually did not afford protection and to determine if one gave them in a prime boost fashion, would it in fact give protection? That was what we now know as the RV144 trial. And again, for the fellows and the younger people in the audience who were not there, the amount of disagreement about whether we should go ahead with this was intense. In fact, there were dueling letters, as it were, to science in January and February of 2004, with some people say that they would doubt whether these immunogens have any prospect of stimulating an adequate immune response, and others on the right-hand side saying, with 5 million new infections each year, the luxury of time is absent, and the ongoing prime boost is scientifically justified, morally correct, and strategically important. It was clearly the epitome, in this particular case, of the conflict between an empiric approach and a theoretical approach. Now, this is the HIV envelope trimer, and we'll get back to the importance of a stable trimer in a moment. The RV144 approach was to essentially take a monomer and empirically vaccinate someone with it without knowing what the response that you would be inducing is, and in fact, would it be protective? The trial went on using this HIV envelope monomer, and there was a 31% efficacy, not ready for prime time, but clearly the first signal of efficacy in any HIV vaccine trial to that point. When one went back and looked at the correlates of protection, correlates of risk analysis, it became clear that antibodies to the V1, V2 glycan region were critically important. In fact, the insights we got from RV144 was that IgG antibodies against the V1, V2 were associated with reduced infection. These were not neutralizing antibodies. Monoclonals that were made against the particular epitope that was targeted by these non-neutralizing antibodies mediated ADCC activity, bringing up another potential mechanism besides neutralizing antibodies. And then the interesting observation that IgA antibodies had specificities that actually blocked the IgG-mediated ADCC. So the strategies now to follow up is to amplify the RV144 response, namely try to increase its strength, its breadth, its durability by multiple boosts, different and modified vectors, and the addition of adjuvants. In fact, that approach, which has been referred to now as the Pox-Protein Public-Private Partnership, involving NIAID, the Bill and Melinda Gates Foundation, and others, will be embarking on two separate trials. One is a trial for the licensure of the products shown on this slide, and the other is a research track to assess the impact of different approaches, different priming, alternate doses, and different vectors and adjuvants. And that is now the approach that is being taken as a follow-up of the trial. While we continue to pursue this research agenda, the scientific community has agreed that novel approaches are needed. To that end, in this science article, we described a shift in research focus to less product development and more vaccine discovery research, which, as it says in this quote from the paper, will require a more nimble, robust, and expandable and contractible clinical research infrastructure, but also will require new ideas. In essence, we were going into the theoretical approach towards vaccine development. The theoretical or deductive approach towards HIV vaccine development was heavily predicated on the ability or not to induce broadly neutralizing antibodies, which classically are protective antibodies in other diseases. Now, before we get into a discussion of that, there are a couple of important issues regarding neutralizing antibodies in HIV-infected individuals. First, broadly neutralizing antibodies that are detectable in plasma are elicited in really only a very minority of individuals, about 20%, and these appear only after 2 years or longer of infection with ongoing active virus replication. Most of these antibodies demonstrate a high degree of somatic mutation, indicating the long-term response to active virus replication, and certain of these broadly neutralizing antibodies have other unusual traits, such as autoreactivity. So it was an unprecedented issue here regarding these very unusual neutralizing antibodies. But these individuals who did develop neutralizing antibodies, these antibodies were made into monoclonal antibodies that were used, as shown on this schematic diagram of the HIV trimer, were able to bind to specific sites on the envelope, such as the CD4 binding site, the membrane proximal external region, the V1, V2 glycan region, all of these as shown on this slide. So we now, using the envelope trimer as the target here, we were able to delineate the potential epitopes that we want to make antibodies against and that we might ultimately use in inducing antibodies. But there was some real critical challenges in HIV vaccinology that we had to address based on the induction of these antibodies. First, do these antibodies actually offer protection against acquisition of HIV infection? You wouldn't want to induce them if in fact they were not protective. And if they did protect, can these antibodies actually be induced by vaccine immunogens? So let's address the first question. Do they protect? We, the field, are now in the process of approaching it from a number of standpoints. One is the actual passive transfer of broadly neutralizing antibodies, and the other is the use of gene therapy or gene-based vector-based approaches of genes that encode broadly neutralizing antibodies. There's kinds of studies that David Baltimore, Phil Johnson, and a number of other investigators have been approaching. And in fact, if you look at the history of this, going from the first generation of neutralizing antibodies, passive transfer has actually been shown to protect macaques against challenge. So the protective aspect. This was extended in subsequent studies using the next generation of monoclonal antibodies that I just alluded to on the previous slide. And then one asked the question, if it can protect against infection, can it actually suppress viremia? And studies shown here, and these are just representative, a number of labs have done similar studies from Michelle Nutzenzweig and Dan Baruch and Mal Martin and others, showing that actually you can get a therapeutic effect from neutralizing antibodies that are passively transferred into the monkey model. In our own lab a few months ago, we used a panel of broadly neutralizing antibodies to determine if these can inhibit the entry into CD4 positive T cells of a virus that was isolated from the reservoir and whether or not these antibodies can actually suppress the reservoir. And as you've shown on the slides here, the red triangle is the control IgG, whereas the others are the individual monoclonal antibodies like PG16, PG9, VRC01, etc., all quite effective in suppressing the replication of reservoir virus in an individual. The other question is, if you have active viremia, can you suppress it? And in some preliminary studies that were done in anticipation of larger studies, these are data from Julie Ledgerwood showing that the passive transfer of VRC01 in individuals with various levels of plasma viremia were able to be temporarily suppressed, as shown on 4 out of 5 of these subjects, in which the effect was quite striking, particularly in subject number 3. So now, these type of passive transfer studies can also serve as a guide to future vaccine regimen design. Namely, what is the breadth of these antibodies against a range of virus isolates? What is the minimum titer that you would need to achieve in a vaccine to get the protective effect that you saw with the passive transfer? And finally, you can identify what the most appropriate envelope epitopes might be for immunogen design. Now comes the critical question, which everyone is excited about and in some respects a bit frustrated by. Can broadly neutralizing antibodies be induced by vaccine immunogens? A clue to the approach of this was the natural infection and a study reported a couple of years ago by Bart Haynes, in which he studied carefully the coevolution of the mutation of a natural infection virus and the evolution of an antibody in an HIV-infected individual over time, accumulating more mutations, longer CDRH3, to ultimately result in a broadly neutralizing antibody that didn't neutralize the virus in this patient, but broadly neutralized against multiple other isolates, prompting a model that was very well articulated in a paper in Science from 2013 by Michelle Nutzenzweig and colleagues, in which an immunogen design might mimic this coevolution that Haynes and colleagues had put forth in their paper, namely do an iterative immunogen design to engage the B-cell lineage along the way to push more towards greater mutations, longer CDH3, and ultimately the effect that was seen in the naturally infected individual. And so the challenge summarized on this slide is, how do you get those neutralizing epitopes that I alluded to before to be put into the kinds of immunogens that can induce a broadly neutralizing antibodies? I want to quickly summarize what that pathway would be by taking a look at a putative B-cell pathway to an HIV vaccine with target structure engagement and then ultimately, hopefully, the induction of a broadly neutralizing antibody. First, the target we know, as I mentioned, is the trimeric heterodimer of HIV envelope. The structure is very important. The ability to now have in our hand a stable, soluble trimer of HIV based on the work of a number of laboratories, Ian Wilson, John Moore, Dennis Burton, Peter Kwong, and others, have now put us into a position to have that soluble structure that would be so important in engaging the B-cell repertoire. Once you have it, you want to engage the naive B-cell repertoire, the germline approach, which would be hopefully in a situation where we would be able to get that iterative approach and get the kind of evolution from germline to mutated to the kind of high affinity antibody with increased breadth, ultimately resulting in a broadly neutralizing antibody. And so over this talk, I have described for you, both from a historical standpoint and from a present approach to HIV vaccine design, both the empirical or inductive approach, as well as the theoretical or deductive approach. And I hope it's become clear to us all, because I am certainly convinced that towards an HIV vaccine would actually be a combination of both. We must pursue the theoretical or deductive approach, and sooner or later we would have to take the empirical step. The one thing that is critical here is, in fact, that we do not have an option for failure. Because as I've said on a commentary that I wrote a year or so ago, that in order to end the HIV/AIDS epidemic in a sustained way, despite the fact that prevention modalities in the non-vaccine arena have brought us a long way, if we are really going to put a sustained end to the epidemic, we will absolutely need an HIV vaccine. And so once again, I want to thank you very much for giving me the opportunity to be with you here by video, and I wish you a successful and fun meeting. Thanks again.